Okay, so this begins chapter 6 on tissues and membranes. We're going to talk about the four major kinds of tissues, and we're going to talk about um, how we can recognize them under the microscope and what it is they do in the human body. Okay? We'll also talk a little bit about what membranes are and what types of membranes there are in the body and where they're found and what they do. Okay? So tissues are the next level of organization up from cells. Remember that cells are the smallest unit of living matter, and tissues are groups of cells that have similar characteristics that act as a team to do something that a single cell couldn't do on its own. The study of tissues is called histology, and it requires the use of a microscope. And the four major types of tissues are epithelial tissue, which forms coverings and barriers and engages in secretion and absorption and protection of underlying tissue. Connective tissue, which falls into three broad groups: liquid connective tissue, such as blood and lymph; structural connective tissue, such as cartilage and bone; and then connective tissue proper, such as dense regular connective tissue, dense irregular connective tissue, reticular connective tissue, adipose tissue, and loose connective tissue. Nervous tissue, which is designed for sending and receiving electrical information, is found in the brain, the brainstem, spinal cord, and the nerves. And then muscle tissue, which is capable of turning chemical energy into the energy of movement. And there's three types: cardiac muscle tissue in the heart, skeletal muscle tissue connected to our bones, and then smooth muscle tissue, which is in the walls of our organs, our blood vessels, our lymphatic vessels, etc. Okay? Of the three, only skeletal muscle tissue is under our conscious control. The other two tissue types either respond to chemicals called hormones or to certain reflex actions that are caused by sets of conditions such as stretching, or by a part of the nervous system that works without us having to consciously control it, called the autonomic nervous system. So here's a little bit on epithelium.
Epithelia form the surface layer of the body, line body cavities, hollow organs, and structures, and constitute most gland tissue. Any substance that enters or leaves the body must cross an epithelium. All epithelia consist of one or more layers of tightly packed cells and little or no extracellular matrix between cells. Distinguishing features of an epithelium include a free apical surface and a fixed basal surface. Specialized intercellular junctions join adjacent cells, and a thin basement membrane separates the basal surface from underlying connective tissue layers. Although epithelia lack blood vessels, many sensory nerve endings are present. Epithelia have a high regenerative capacity that allows them to maintain their function. The primary functions of an epithelium are to resist dehydration and injury from physical, chemical, and biological agents, to selectively regulate materials entering and leaving the body, to secrete products produced by epithelial glands, and to monitor the environment through specialized sensory nerve endings and specialized epithelial cells. Epithelial tissues are classified using two basic criteria: the number of cell layers and the shape of the cell at the apical surface. An epithelium composed of one layer of cells is termed simple. If there are two or more layers of cells, the epithelium is termed stratified. When the apical cells are flat and thin, the epithelium is classified as squamous. Apical cells that are somewhat square shape belong to a cuboidal classification, while those with a rectangular shape are termed columnar. By combining the criteria of cell shape and number of layers, all epithelia can be classified easily. For example, A simple squamous epithelium is composed of a single layer of thin, flat cells. In contrast, a stratified squamous epithelium is composed of multiple layers of cells, with the apical surface layer being thin and flat. These same tenets could be applied to cuboidal and columnar epithelia. There are two special types of epithelia. Transitional epithelium, found in the urinary tract, is a stratified epithelium that gives the impression of transitioning from cuboidal, when the epithelium is relaxed, to squamous when it is stretched. A pseudostratified epithelium, found in the trachea, for example, is a simple epithelium in which all cells contact the basal lamina, but only some reach the apical surface. Cells are of varying heights, and their nuclei are at different positions in the epithelium, giving the impression of stratification. Some epithelial classes have specializations of the free surface of apical cells. Simple columnar epithelial cells in some organs have cilia, which are small, finger-like motile projections. Thus, simple columnar epithelium is subclassified as ciliated or non-ciliated. In parts of the digestive and urinary tracts, epithelial cells involved in absorption have a dense collection of short cytoplasmic processes called microvilli on their free surface. Microvilli are non-motile and serve to increase surface area. Because the microvilli appear fuzzy in the light microscope, cells with numerous microvilli are said to have a brush border. A stratified squamous epithelium has either dead or living cells as the surface layer. The dead cells contain the protein keratin. Therefore, stratified squamous epithelium is subclassified as keratinized or non-keratinized. Epithelial cells are tightly connected to adjacent cells by specializations called intercellular junctions. There are 4 types of junctions: tight junctions, adhering junctions, desmosomes, and gap junctions. Tight junctions, also known as zonula occludens, are bands of fused cell membrane near the free surface of adjacent epithelial cells. These junctions attach a cell to its neighbors, encircling cells and sealing the space between them. Thus, substances crossing the epithelium must pass through epithelial cells, not between them. Tight junctions are essential to the primary function of an epithelium, which is to regulate substances that enter and leave the body. Adhering junctions, also known as zonula adherens, have a similar band-like arrangement seen in tight junctions. They are located deep to the tight junctions and have numerous microfilament proteins from the cytoplasm that act as anchors for adjacent cells. There is a small gap between the cell membranes of adjacent cells at adhering junctions. Desmosomes, also known as macula adherens, are small button or snap-like junctions. These form at the points of mechanical stress. They are different from tight and adhering junctions in that they do not encircle the entire cell. Gap junctions are points where small pores connect the cytoplasm of adjacent cells. The gap junction consists of a series of hollow bridging structures called connexons. Small solutes, such as ions, glucose, and amino acids, pass through connexons from the cytoplasm of one cell into a neighboring cell.
So, as we noted in the video, right, these are cells that are tightly connected to each other. They form coverings, so coverings of the insides of hollow organs, coverings of the external surface of the body, and coverings of pathways that are open to the outside world, such as our respiratory tract and our digestive tract. It is going to line and protect most of those surfaces. In some cases, epithelial tissue is going to have multiple layers, And in that form, it's primarily protective and forms a physical barrier so that it's more difficult to get infection, it's more difficult to have physical trauma, and so on. And in some places, the epithelial tissue is going to be one layer of cells, and that's primarily for processes like secretion and absorption. Okay? So, epithelial tissue can transport material either through these teams of cells or between these teams of cells. It's found lining the lower respiratory, the lower digestive, okay? It's also found in places like the kidney, okay, where secretion and absorption take place. The absorption function is to pull material from what we call the apical side of the cells, the side that faces the free space, and then to move that material through those cells to the other side. And so the cells determine what goes through. Filtration is another property of simple epithelial tissue, and essentially filtration is a barrier that has openings in it, and what gets through the openings is anything tinier than the openings. Okay, so filtration, as an example, in humans happens in the kidney. You do this every day when you make coffee from ground coffee beans, right, with the coffee filter. Secretion is where the cells manufacture something inside them and they spit it out. Okay, we noted in the chapter on cells that one of the ways this happens is through a process called exocytosis, which is a type of active transport. And then transportation, essentially, that's moving material from one location to the other, usually across the apical surface. An example of this would be the ciliated epithelia that lines the trachea that moves mucus up towards the oral cavity so that you don't end up swallowing this sticky secretion that has bacteria and particulate matter in it. Okay? So now we want to ask the question, when we look at epithelial tissue, what do we see? Well, its purpose is to form barriers and engage in secretion and absorption. The outer surface is usually going to face either the lumen of an organ or a free surface. And then the basal surface is attached to underlying connective tissue. The tissue itself is avascular, meaning no blood vessels. And so in order to get nutrients, it relies on diffusion from the connective tissue that lies beneath it. And that's one of the reasons why in stratified epithelial tissue, which has more than one layer, the top layers have less access to the nutrients and the oxygen than the deeper layers do, and they either die and are shed off or are more easily disconnected from the underlying layers, whereas simple epithelial tissue is always in relatively close contact with the blood supply. We can class epithelial tissue according to the shape and the number of layers. The shapes include squamous, which is pancake shape, cuboidal, which is box shape, and columnar, which is post shape. Simple epithelia is a single layer of tissues, so you can have simple cuboidal or columnar or simple squamous, and you can also have a particular type of simple epithelial tissue called pseudostratified ciliated columnar epithelium. Stratified epithelium is multi-layered. Examples include transitional epithelium as well as stratified squamous and stratified columnar and cuboidal. Stratified columnar and cuboidal are much rarer to find in the body than transitional. So where do we find each of these? Simple cuboidal, you're going to find in places like the kidneys and the sweat glands. Simple columnar, you're going to find in the digestive system. Pseudostratified ciliated columnar, the nasal cavity and the trachea. Transitional epithelia is going to be found in places like the bladder and the ureters, which are the pipes that bring urine into the bladder from the kidneys. And stratified squamous epithelium, if it's not keratinized, i.e. it's not dry, is going to be found in the mouth, the rectum, the anus, and the throat. From the area of the throat behind your mouth, which is called the oropharynx, down to the area of the throat behind your voice box, which is called the laryngopharynx. Okay? Transitional epithelium is special in that it has the ability to stretch and snap back, and that's why it tends to be found in places like the urinary system. The other tissues, when they're present as simple tissues, are primarily there for transport purposes or secretion purposes. When they're stratified tissues, they tend to be there for protection, okay, because it's tougher stuff. Glandular epithelia's job is to serve as a secretory barrier, okay? There will be exocrine and endocrine glands embedded in the glandular epithelia. They'll be surrounded by an epithelial layer like you can see here. Examples of exocrine glands include sebaceous glands, such as the one seen here, which produce sebum, which is secreted onto the surface and keeps the hair follicle flexible and the skin supple, and also produces an antibacterial environment, so it cuts down on the potential for infection. This is an apocrine sweat gland or sudiferous gland, and it produces a lipid-rich sweat that when bacteria act on it produce body odor, and these guys really are not critical to our survival, but the eccrine sweat glands are important in regulating our body temperature, because when we get hot, they create sweat by filtering blood, and that sweat pools on the skin, and as it dries, that cools the skin, and that cools the blood, and that turns out to cool the body. Okay. Note that an exocrine gland is always defined by the fact that it has a duct, a pipe, through which the material is secreted, and that differs from endocrine glands, which are found scattered throughout the body. Examples include the hypothalamus up underneath the brain, the pituitary gland under the hypothalamus, the thyroid gland in front of your voice box in your throat, the parathyroid glands, which are right behind the thyroid, the adrenal glands, which are on top of each of your kidneys, and your pancreas, which is wedged between the first part of your small intestine and an organ called your spleen, and it sits behind the stomach. The thymus, which sits in front of your heart and behind your sternum, your breastbone, and your gonads, which in guys are in a sac called the scrotum, and in females are inside the pelvic cavity, and they're known as ovaries. Okay, and guys are called testes. And what they do is they actively transport materials that they manufacture right into the bloodstream for the purposes of controlling the body's homeostasis. Okay, and these chemicals are known as hormones. So let's take a look now at connective tissue.
Connective tissue is diverse, abundant, and widely distributed through the body. Its primary functions include structural support, binding structures together, fat storage, exchange of nutrients and metabolic waste, and defense and protection. All connective tissues share three basic components: cells, protein fibers, and ground substance. Together, the protein fibers and ground substance form the extracellular matrix. The proportions of these components, in addition to different cells and protein fibers, help define the various categories of connective tissue. In the adult, connective tissue can be divided into three broad categories: connective tissue proper, supporting connective tissue, and fluid connective tissue. There are two basic subcategories of connective tissue proper: loose connective tissue, and dense connective tissue. Loose connective tissue has abundant ground substance with scattered cells and loosely arranged protein fibers. By comparison, dense connective tissue has abundant, tightly packed fibers but relatively few cells and little ground substance. Loose connective tissue can be subdivided into areolar, adipose, and reticular connective tissue. Areolar connective tissue is found in the subcutaneous layer and around organs. It has a gel-like ground substance, numerous blood vessels, and scattered cells known as fibroblasts. Adipose connective tissue is found in subcutaneous tissue and around some organs. It is composed of closely packed adipocytes or fat cells. Reticular connective tissue forms the stroma, a connective tissue framework, of lymph nodes, the spleen, the thymus, and bone marrow. It is composed of a network of branched collagen fibers, known as reticular fibers, with scattered fibroblasts and leukocytes, or white blood cells, embedded in a gel-like ground substance. Dense connective tissue can be subdivided into dense regular, dense irregular, and elastic connective tissue. Dense regular connective tissue forms tendons and most ligaments. It is composed of densely packed parallel collagen fibers with fibroblasts between the layers. There is little ground substance. Dense irregular connective tissue is found in the dermis, periosteum and perichondrium, and the capsule of organs. This type of connective tissue is composed predominantly of randomly arranged collagen fibers with fibroblasts interspersed. There is more ground substance than found in dense regular connective tissue. Elastic tissue is found in the walls of elastic arteries, the trachea and bronchi, and the vocal cords. This type of connective tissue is composed predominantly of branching elastic fibers, with fibroblasts in some spaces between the fibers. Elastic fibers, formed by the protein elastin, stretch under tension and then return to their original shape when tension is released. Supporting connective tissue can be subdivided into cartilage and bone. Cartilage has a semi-solid extracellular matrix that allows flexibility, whereas the solid matrix of bone is more rigid and provides greater support. There are three types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage is the most common type. It is found on the articular surfaces of synovial joints. It also forms the cartilage of the larynx, trachea, and nose. Chondrocytes, or mature cartilage cells, are found within small spaces called lacunae that are scattered irregularly in the extracellular matrix. Collagen fibrils are present within the matrix but not visible in the light microscope. A perichondrium, a dense irregular connective tissue sheath, overlies most cartilage. Elastic cartilage is found in the external ear and in the epiglottis. It contains abundant elastic fibers, which form a web-like mesh around lacunae that contain chondrocytes. Like hyaline cartilage, elastic cartilage usually has a perichondrium. Fibrocartilage is found in intervertebral discs, the pubic symphysis, and the menisci of the knee joint. It contains numerous irregular bundles of collagen fibers. The sparse ground substance contains chondrocytes within lacunae that are frequently arranged in parallel rows. Unlike hyaline and elastic cartilage, fibrocartilage does not have a perichondrium. There are two forms of bone or osseous connective tissue: compact bone and spongy bone. Compact bone usually forms the hard outer shell of bones. Concentric rings of bone, called lamellae, form cylindrical structures known as osteons. Each osteon has a central canal that connects to a network of tiny branching passageways called canaliculi. The central canal and canaliculi contain blood vessels. Osteocytes, or mature bone cells, occupy lacunae, small spaces, between neighboring lamellae. Spongy bone, also known as cancellous bone, usually forms inside of bones. It does not contain osteons. Slender bony plates, called trabeculae, form a bony network. The spaces between trabeculae contain bone marrow and blood vessels. Osteocytes are scattered throughout the trabeculae. The combination of compact and spongy bone allows bones to be both strong and lightweight. Fluid connective tissue is represented by blood. The formed elements of blood, specifically its cells and cell fragments, include erythrocytes, or red blood cells, leukocytes, or white blood cells, and platelets. The watery ground substance of blood contains dissolved protein fibers.
Okay, so the functions of connective tissue are pretty broad, right? It's important for attaching tissues to each other. That's its function in connection. It provides a support structure for the soft tissues of the body, as in bone and cartilage. It protects a lot of our internal organs. Bone and cartilage do that, as well as fat. And you could also argue for some connective tissue proper playing that role. Fat storage is the function of a type of connective tissue called adipose tissue. And transportation is a function of connective tissue such as blood and lymph, okay, moving things around the body. So connective tissue really falls into three broad classes, okay? There's connective tissue proper, which includes dense, regular, and irregular connective tissue, and loose connective tissue and adipose tissue. There's liquid connective tissue like blood and lymph. And then there is also reticular connective tissue, which is also connective tissue proper. And then finally there's structural connective tissue, such as bone and cartilage. Okay, so it falls into those three broad classes. But every time you look at connective tissue under the microscope, one of the things that jumps out is the fact that there's a lot of material between the cells, which is called extracellular matrix, and relatively little material within the cells themselves. Okay, so the cells are widely spaced out. And the extracellular matrix could be a liquid or a solid or a semi-solid. Okay, all depends on the tissue type that we're talking about. Most of it has an adequate blood supply, with the exception of tendons, ligaments, and cartilage. So tendons and ligaments are made of dense, regular connective tissue, and cartilage is its own structural connective tissue. And the way that those tissue components, the cells within those tissues, get their nutrients, again, is largely through diffusion. Okay, because cartilage and ligaments have virtually no blood supply, they tend to be relatively slow healing, okay? And if ligaments are torn, they usually have to be surgically repaired. And if cartilage is torn, there really is no way to regenerate it or to produce a substitute for it, because it's very difficult to get cartilage to stick to bone for a variety of reasons. Tendons have a slightly better blood supply and can heal over a series of weeks if they're torn, or if they're partially ruptured. The protein in the extracellular matrix varies depending on the tissue type. In connective tissue proper, such as loose connective tissue, which is found under all epithelial tissue layers, protein includes collagen and elastin and reticular fibers and keratin in many cases, whereas in adipose tissue it's primarily Really not an extracellular matrix as much as it is fat droplets that are within the fat cells. And in bone, it's a hard matrix called hydroxyapatite, which is calcium phosphate. There's also collagen in there, and there are glycoproteins in there as well. In collagen, not in collagen, in cartilage, it's mostly glycoproteins. There is in some cartilage, like elastic cartilage, elastic fibers, and in fibrocartilage, there are things like collagen fibers. Um, Fibrocartilage is found in places like the intervertebral discs and the meniscus of the knee, and an area in front of the pubic um, uh, bone in your hip called the pubic symphysis. And elastic cartilage is found in your epiglottis in your ear, your external ear. And hyaline cartilage is pretty much in all the other collagen locations. The elastin provides the ability to stretch and snap back. The collagen provides resistance to twisting. And the reticular fibers, where they're found, are usually a framework for other cells to sit on top of. So they're sort of like floors in a building, if you like. The different types of connective tissue shown here, right? Loose connective tissue, including areolar, adipose, and reticular connective tissue. That's connective tissue proper. Dense, irregular, and regular connective tissue is also connective tissue proper. And again, it's found primarily in joint capsules, ligaments, tendons, and around certain organs. And cartilage, hyaline cartilage, is found at the ends of bones that articulate as joints, come together to form joints, so it pads and protects them. It's found in the upper respiratory system where it holds your voice box and your trachea open. Fibrocartilage, we've discussed where that's found, and elastic cartilage as well. Bone, of course, is the primary component of the skeletal system, and it comes in two forms, compact bone and spongy bone. But the extracellular matrix is mostly calcium phosphate, and then collagen, and then some glycoprotein. And then finally blood and lymph, where the extracellular matrix in blood is called plasma, and in lymph it's simply lymphatic fluid. Nervous tissue is different yet again. Okay? Nervous tissue is designed to send and receive electrical information through the use of either direct connections between adjacent nerve cells called neurons, or through chemical signals that produce electrical signals when they land on the membrane of the nerve cell. So there's two broad populations of cells in nervous tissue. There are the neurons that do the electrical work, and that's why they have all these extensions coming off of them. And then there's the glial cells, which sort of help the neurons out because they're so busy doing the electrical work that they really don't have time to do the stuff that a lot of other cells do typically. The short extensions in neurons are called dendrites, and they receive chemical signals that produce electrical signals in the membrane. And then this long extension is called an axon, and he sends the electrical signal to the end of the cell, and that results in secretion of a chemical called a neurotransmitter, and that ends up attaching to the next cell and nerve cell in the pathway, or maybe to a muscle cell, and that causes an electrical signal to be produced on that target. Okay, neurons don't divide. Okay, and neurons in the central nervous system don't even repair themselves, and that's why damage to nerve tissue in the central nervous system is pretty much permanent. Neurons in the peripheral nervous system, like in your peripheral nerves, which are the wiring between your brain, brainstem, and spinal cord, and the rest of your body, can sometimes repair itself if the axon gets clipped. It can sometimes regrow and then find its target again. The glial cells help the neurons out, and in the central nervous system, the glial cells include the microglia, which are like the emergency med techs of the central nervous system. They eat up pathogens and they clean up dead tissue. The astrocytes, which attach to the capillaries that run through the central nervous system and also to the neurons, and they basically provide the fuel for the nerve cell, and help with distribution of ions, and the oxygen, of course, diffuses directly through the blood supply and into the fluid that bathes all the nerve tissue, which is called cerebrospinal fluid. And then you've got, in the central nervous system, funny-looking cells called oligodendrocytes, and what they do is they wrap little cytoplasmic extensions that secrete an insulating molecule, which is called myelin, around the axons as a way to both insulate the electrical signal so it doesn't leak out, but also to speed the electrical signal up pretty dramatically. In the peripheral nervous system, satellite cells cover the cell body of the neuron and protect and nourish it, and then Schwann cells become the myelin sheath that protects the axon and speeds up the electrical signal. The glial cells can divide, and they are responsible generally for what we think of as brain tumors, right? Whereas the neurons, essentially, you get all the neurons you're going to get when you're born, and as you get older, they just die off. And that's one of the reasons why, as we get older, it's harder to learn things, it's harder to remember stuff, it's harder to perform actions, because that wiring is beginning to break down.
The nervous system is a network of specialized cells that monitors the internal and external environment, processes information, and initiates commands through which the body reacts. Nervous tissue is composed of two types of cells: neurons, or nerve cells, and glial cells. A neuron, the basic functional unit of the nervous system, is specialized to conduct electrical impulses. Neurons have several important functional properties: a high metabolic rate, most are incapable of cell division, and they typically function throughout life. The size and shape of neurons is variable, but they share basic features. The cell body, or soma, receives, integrates, and sends nerve impulses. It typically contains a large nucleus that often has a prominent nucleolus. Ribosomes are abundant and form clusters known as chromatophilic substance, or Nissl bodies, that stain darkly with basic histological dyes. Two types of processes project from the cell body: axons and dendrites. Axons direct information away from the cell body. Most neurons have a single, long, thin axon connected to the cell body at a region known as the axon hillock. The axon generally does not branch, but it may have occasional side branches called collaterals. Most axons terminate as multiple fine branches called telodendria. These have distal expansions known as synaptic knobs, also called end bulbs or terminal boutons. Dendrites direct information toward the cell body. They are relatively short, tapered processes that branch along their length. They often possess small, irregular projections called dendritic spines, where axons can synapse. A synapse is where information is transferred between nerve cells, or to an effector cell, or to a sensory receptor cell. Glial cells, which are also known as neuroglia, are support cells that represent about 60% of cells in the nervous system. Four types of glial cells are found in the central nervous system, or CNS: astrocytes, ependymal cells, microglia, and oligodendrocytes. Two additional types of glial cells are found in the peripheral nervous system, or PNS: Schwann cells and satellite cells. Astrocytes are the most abundant type of glial cell in the CNS. They are star-shaped, with numerous branching processes whose expanded ends contact and support neurons. In some astrocytes, these processes contact capillaries as perivascular feet that help form a blood-brain barrier. This regulates the passage of substances between the CNS and bloodstream. Other important astrocyte functions include regulation of the chemical environment and promotion of neuronal development. Unlike neurons, astrocytes are capable of cell division and can replace dying neurons with a glial scar. Ependymal cells are epithelial cells that line the ventricles of the brain and central canal of the spinal cord. They play an important role in the production and circulation of cerebrospinal fluid, or CSF. Microglia are small elongated cells that have short, irregular processes. Their main function is to respond to infection in the CNS. When activated, they become phagocytic and remove cellular debris associated with damaged nervous tissue. They also secrete immunoregulatory substances, such as cytokines, as part of this response. Oligodendrocytes have large cell bodies with thin, flat processes that wrap repeatedly around portions of axons in the CNS. This wrapping process produces an insulating cover known as a myelin sheath. Each oligodendrocyte can myelinate multiple axons. However, each axon requires multiple oligodendrocytes for complete myelination. Two additional types of glial cells are found only in the PNS: Schwann cells and satellite cells. Schwann cells, also known as neurolemmocytes, myelinate PNS axons. Each Schwann cell forms a myelin sheath around a single axon, unlike oligodendrocytes in the CNS that can myelinate multiple axons. As with oligodendrocytes, multiple Schwann cells are required to myelinate each axon. Satellite cells are small and flat. They provide physical and chemical support for neurons located in ganglia.
Muscle tissue is composed of fibers that contract to cause movement of a body part. The three types of muscle include skeletal, smooth, and cardiac. Skeletal muscle is multinucleate rod-shaped cells that form bundles called fascicles, and then bundles of fascicles form the muscle. And there's connective tissue throughout holding all this together, and that attaches to bone, and we consciously control it. So we say that it's striated because it has the appearance of light and dark bands running through the muscle tissue, and we also say that it's voluntary because we control it with our conscious thought. The light and dark bands, the striations, are just areas where the proteins that make the muscle contract overlap each other. In the areas where there are dark bands, that's where the contractile proteins actin and myosin basically sit on top of each other, and in the light areas, that's where they don't. Okay, and so when muscle contracts, what happens is that the actin and the myosin slide over each other more extensively, and the dark bands get thicker, and the light bands get thinner, and the whole muscle will contract. Smooth muscle is found in the walls of hollow organs and blood vessels, and attached to every hair in your body, and in the iris of your eye, okay, among other places. And it's called smooth because it doesn't have any striations. It's not under conscious control. It's operated by something called the autonomic nervous system. But it does respond also to hormones and to local reflexes. And then cardiac muscle is striated and involuntary, just like smooth muscle is involuntary, but it is also not under conscious control, okay, and it's only found in the heart. And it has one very special property that separates it from all the other muscle types, which is it is autorhythmic, meaning it can produce its own electrical signal that results in the entire muscle layer contracting together. And that's what makes it a really good choice for tissue to use as a blood pump. So you can see the different types of muscle tissue here and what they look like under the scope. Okay? And they're all designed just to turn chemical energy into the energy of movement.
Muscle tissue is widely distributed throughout the body. The primary function of muscle tissue is to produce movement of the skeleton or other tissues of the body. This results in body or organ movement, maintenance of posture, communication such as facial expressions and vocalization, control of body openings, and heat production. Muscle tissue is composed of cells called muscle fibers. Each fiber contains contractile proteins that contribute to the properties of muscle tissue. These include excitability, conductivity, contractility, elasticity, and extensibility. There are 3 types of muscle tissue: skeletal, cardiac, and smooth. Skeletal muscle is under voluntary control and is usually attached to bones via tendons or other connective tissues. Skeletal muscle fibers, or cells, are long, thin, and cylindrical. Each fiber has multiple oval-shaped nuclei located just deep to the sarcolemma, the plasma membrane of the muscle cell. Skeletal muscle fibers are surrounded by a delicate layer of connective tissue called the endomysium. Multiple muscle fibers are bundled together to form fascicles, which are covered by a dense connective tissue layer called the perimysium. The perimysium contains the nerves and blood vessels that supply the muscle fibers. Numerous fascicles are then collected to form a muscle that is covered by an external sheath of dense connective tissue known as the epimysium. The cytoplasm of a skeletal muscle fiber is called sarcoplasm. The sarcoplasm of a muscle fiber contains long cylindrical structures called myofibrils. Each myofibril is composed of bundles of short myofilaments. There are 2 types of myofilaments: thin filaments, which are composed primarily of the proteins actin, tropomyosin, and troponin, and thick filaments, which are formed by the protein myosin. The 2 types of myofilaments are arranged in parallel, and when viewed along their long axis, they exhibit alternating dark and light bands known as striations. The dark bands, called A bands, represent the entire length of the thick filament. Conversely, the light bands, called I bands, represent thin filaments only. The Z discs, dark transverse bands at the center of the I bands, represent fibrous protein structures composed of connectin, to which the thin filaments are anchored. The sarcomere is the functional contractile unit of skeletal muscle. It is defined as the region within a myofibril between adjacent Z discs. There are numerous sarcomeres in each myofibril. Cardiac muscle is found only in the walls of the heart and is under involuntary control. Cardiac muscle fibers are short branch cells with 1 or 2 centrally located nuclei and numerous mitochondria. These fibers exhibit striations and banding patterns similar to those seen in skeletal muscle fibers. Cell junctions, called intercalated discs, are unique to cardiac muscle. 2 specializations within these junctions link cardiac muscle fibers both electrically and mechanically. Gap junctions form an electrical connection by allowing ions to flow from cell to cell, facilitating the spread of impulses that enable cardiac muscle to contract as a unit. Desmosomes contribute to intercalated discs as mechanical junctions that prevent cardiac muscle fibers from pulling apart during contraction. Smooth muscle is located in the wall of blood vessels, ducts, and organs, and is under involuntary control. Smooth muscle cells are elongated, with tapered ends, fusiform-shaped, and contain a single, centrally located nucleus. The myofilaments are not arranged in any specific pattern, and, therefore, do not produce visible striations or form sarcomeres. Instead of the Z discs present in skeletal and cardiac muscle, Smooth muscle contains dense bodies that serve as attachment sites for thin filaments. Dense bodies are small protein concentrations distributed throughout the cytoplasm. Typically, smooth muscle cells are tightly packed and arranged in sheets. Their contraction is slow and resistant to fatigue. Like cardiac muscle, some smooth muscle fibers contain gap junctions that spread electrical impulses and allow smooth muscle to contract as a coordinated unit.
Tissue repair is something that happens when we damage these tissues, and they can replace themselves either by fibrosis, which is the production of scar tissue to patch a hole in the connective tissue or in whatever tissue we're dealing with, or it can heal by regeneration. Okay, tissues that heal by fibrosis include most connective tissue. In many cases, you'll see nerve tissue heal by fibrosis if it's damaged. And tissues that regenerate include epithelial tissue. The tissue that we find in the liver, for instance, is an example of tissue that can regenerate, and the tissue in the top of your skin, the stratified squamous epithelia, can regenerate. And then muscle tissue depends on the type of muscle tissue. Skeletal muscle tissue tends to scar. Smooth muscle tissue usually heals by regeneration, and cardiac muscle tissue tends to scar. Okay? The problem with a scar is that a scar doesn't have the same function as the tissue that it replaced, and the result of that is loss of tissue function. So what happens with an injury is that chemicals are released from the damaged tissue that usually, through regeneration, cause the production of chemicals called growth factors that promote mitosis and promote the regrowth of the tissue to basically cover and replace the area damaged by the injury so that you restore full function. In fibrosis, it's different. The damaged tissue is replaced by connective tissue, which basically just patches the hole, and the process again is kicked off by chemicals that are released from the damaged tissue. So this is a look at the steps in tissue repair.
Tissue repair is the final phase in the inflammation process from a laceration. The basic methods of repair involve healing by regeneration, or in a deeper laceration, fibrous connective tissue repair known as scar formation. A superficial laceration occurs in the epidermis layer and only slightly into the dermis layer. The damaged tissue will start being replaced by cell division. Epithelial cells begin to regenerate from the edges of the laceration. The new cells mature and fill in the area involved in the injury. When complete regeneration occurs, normal structure and function is restored. In a deeper laceration, the tissue injury involves both the epidermal and dermal layers and will require scar formation to repair itself. The injured area starts to fill with exudate, which is fluid seeping from the capillaries and contains fibroblast cells. These cells are active in the repair of tissue. The epidermal layer begins to regenerate from the sides of the laceration, while the capillaries and fibroblast form new granulation tissue to fill the dermal layer. The epidermal layer heals and fibroblasts develop the permanent scar tissue. The scar is the bridge between the normal tissue and the laceration. It does not restore normal structure and function to the area.
So you can see here the steps involved in tissue repair in, say, the skin. All right, you get a wound and you get bleeding, and that will eventually form a blood clot, and that will lead to the production of a scab, and that's basically a temporary seal while we start the repair process underneath. Now, if the damage goes below the epithelial layer, what will happen is that scar tissue will form, epithelial cells will multiply and grow over it, and you'll end up with a scar covered with epithelial tissue, but which lacks things like hair follicles and sebaceous glands and sudiferous glands. So basically, it's just a patch. Whereas if the damage doesn't go all the way through the epithelial layer, what will happen is that you'll grow back that epithelial layer, and you won't lose any of those other structures that we associate with it. So the reason for this is that if the damage is deep enough, you go through the bottom layer of the epithelial tissue, which is called the basal layer, and that's what's responsible for making the epithelial tissue above it. And if those cell populations are missing, then we can't do that anymore, and we essentially just patch the stuff with epithelial tissue from surrounding areas, but we lose the ability to have those little other features of the skin, the hair follicles and the glands and so on. Okay? Membranes are just sheets of tissue that cover surfaces or line body cavities or surround organs. So examples include cutaneous membranes, such as what covers the outside of your body, okay, your skin. Mucous membranes, which line cavities that are open to the outside world, such as your urinary system, your digestive system, your respiratory system. And then serous membranes that line cavities that aren't open to the outside world, such as the pleural cavities where your lung sits, your pericardial cavity where your heart's located, and your peritoneal cavity, which contains things like your lower digestive system. Okay? Connective tissue membranes include synovial membranes, which line joints and produce a lubricating fluid called synovial fluid that produces friction in the joint, but also nourishes the cartilage at the ends of the bones that are in the joint. The periosteum, which provides nutrients and also bone building and dissolving cells to the bone itself, okay, called osteoblasts and osteoclasts. Perichondrium, which provides a blood supply to the cartilage and also a source of cartilage cells. Meninges, which are lining the central nervous system and basically stabilize and protect the brainstem, and the spinal cord. And then finally fascia, which is connective tissue that attaches the skin to the underlying muscle. You can see here where some of the different membranes are located. Okay? Remember that, again, the job of the membrane, if it's a serous membrane, is to both protect but to lubricate the internal organ and to isolate it from infection. While the mucous membrane produces mucus, and the job of the mucus is to trap pathogens and particulates and help to flush them out of systems that are open to the outside world, and also prevent them from penetrating through those cavities and getting into the blood supply. Okay? While the cutaneous membrane is designed, again, to protect from infection and trauma and chemical insult, and produce an environment that essentially cuts down on the possibility of infection due to the fact that the epithelial tissue is constantly shed and regenerated, and due to the fact that the secretions, sweat and oil, create an environment that flushes bad stuff off your skin, but also creates an environment that they don't like to grow in. Okay? The other membranes, synovial membranes and periosteum and perichondrium, we've discussed in the meninges as well, and the fascia. Okay? The mucous membranes and the cavities that they line and why. Okay? We've discussed that. Serous membranes and their location, right? Pericardium around the heart, peritoneum in the area below the diaphragm, going all the way down to the pelvic region, and the pleural membranes around the lungs. Okay? And again, that's to lubricate and protect those tissues so that they don't rub against each other and produce friction and scarring. Okay? And so that brings us to the end of this podcast. If you have any questions, by all means, contact me during my office hours, and I'll get back to you. And I'll see everybody in the next podcast. Thank you for listening.